Good morning or afternoon to everybody who's tuning in around the world. This is Tom Peterson, and this is our, I believe, 11th in a global online seminar series in biodiversity informatics. I'm very pleased to have you all participating. It's a little disconcerting when I do these broadcasts because I can't see you all, even though I know that there are, on average, 50 or 60 people tuning in via various live windows into these seminars. So I'll introduce our speaker from this month in just a moment, but I will let you know that next month we'll be back on our Thursday morning, 9 a.m. Kansas time schedule. And I'm awaiting a final word from the speaker, but I think you'll like what we have lined up. It should be some nice, very solid marine biogeography work. So I'll announce that very soon. And then also another bit of business is about questions for this seminar. When Dr. Kriticos finishes his seminar, there's always a little bit of a gap before we get your questions in. So I would encourage you to email your questions to me either at my own email address, which is town@ku.edu, or on the biodivtraining@gmail.com address. But I would urge you to send the questions when they come to your mind, and don't wait until the end of the seminar. And that way we'll have some questions waiting for Dr. Kriticos. So today I'm very, very pleased to have as our seminar speaker and guest Dr. Darren Kriticos. He is a principal research scientist at CSIRO in Australia. Indeed, that's why we have this non-standard time, because our usual time would be very early in the morning in Australia. Dr. Kriticos is an ecologist who has, over the years, worked with many of the major biodiversity informatics initiatives that have come through, beginning with Bioclim and GARP, two very early niche modeling algorithms. He's now one of the leaders in development of the software for CLIMEX and DIMEX. Very generally, he's interested in the ecological basis of niche models. Most recently, or recently I should say, I don't know the most recently, Dr. Kriticos has been part of a team that has produced CliMond, which is a new global climate data set usable for many applications in biodiversity informatics and in other fields. And very nice thing about this seminar is that for the first time, this is a seminar by request, which is to say one of our participants in Africa, in Tanzania, saw the publication or the website and contacted me and said, can't you come up with a good seminar about CliMond? It sounds very interesting. And so Dr. Kriticos was quite kind to accept the invitation to give a seminar on request. So I won't take up any more of the time than this, but again, do remember to send your questions for Dr. Kriticos as early and as often as you are able, so that we can do those live at the end of the seminar. So, Dr. Kriticos, I'll send it over to you.
Okay. Thank you very much, Town. Thank you for the invitation. Thank you, Clement, for asking me to give this seminar. It is very early in the morning, so please bear with me. Okay, I'll try and share my screen. Oh. There we go. Town, you have that?
Yes, you are good.
Okay. Okay. So the sorry, I'll just get. Here we go. So CliMond. So this is a background to the um, what is not just a website or a data set. We're a bunch of scientists contributing our time and energy to go and package up, I guess, the tools and data sets that we know are useful to the bioclimatic modeling community. So it's also a collection of documented global climate products and utilities. It's a repository for the definition of the Bioclim variables. It's a fairly new addition. And it's also this website to go and distribute the data and utilities. So CliMond is, if you're wondering where it comes from, it's derived from the French climat mondial, because WorldClim was already taken. Okay, we aim to document all of the material that we put up on the site so that it is instantly citable and comprehensible, that you can understand what decisions we had to go and make along the way to go and provide those products. So who we are, we're this small group. We don't have an official organizational structure. It's just people coming together, and we certainly welcome anyone to come along and contribute with us. You're most welcome. Just contact us. Okay, so why did we create CliMond? This was a question that was thrown to me. It wasn't created so much as it evolved. We needed reliable climate data for our purposes, and we needed a globally conformal data set that would allow us to both compare correlative model behavior with climax models. So we needed data that had the same basis but was in different formats. And we also wanted to go and explore climate change scenarios and model sensitivities. We also needed better ways to go and suitably define our models, and we needed tools to better interrogate them. So some historical context. It helps to go and remember that the gridded climatologies that many of you in your professional lives may have come to take as for granted are all derived from baseline climate station data. You can see in the top map there, that's the distribution of meteorological stations at the time, and you notice that it's quite uneven. That's got implications for just how far you should downscale logically. Now, the computer-based, I apologize that the next few slides are going to be very word dense. The computer-based niche modeling and species distribution modeling were developing in parallel with routines to go and take that station data and interpolate it in a sensible way. So that wasn't an accident. It happened because the people who were developing Bioclim then had on board a team member, Mike Hutchinson. He's a climatologist and geospatial modeler, and he was working with them while I created Bioclim, which later became ANUClim. Now the interpolation routine was extremely clever. And what it did is it made use of covariates to go and estimate climate at locations on a regular grid. Now the Hutchinson spline was included in ArcInfo, and yes, it is extremely powerful, and it is easy to get a result, but it also takes a lot of experience and a depth of knowledge about the climatology and what the drivers of climate in order to create reliable results. And I think we've seen some sort of fairly uneven sets of results. So Mike Hutchinson himself has largely confined most of his involvement in generating Australian data products and I believe a South African data product in terms of these splines. And that they are extremely good quality. Okay, so in order to go and meet our challenges, there were two main contenders for gridded global climatologies. And remember, at this stage, we weren't looking to go and do something ourselves. We were looking to go and buy something off the shelf. So there was WorldClim and the Climatic Research Unit, or CRU, from the University of East Anglia. But each of those had problems. Okay, so the CRU, I believe, produced the world's first gridded terrestrial climatology at 30-minute resolution. Now they had was it Tmin, Tmax, precipitation, RH, solar radiation. They were the variables that we were most interested in, and a series of other variables like frost days and snow duration. That was well documented. And for many years, that 30-minute climatology was the mainstay of our bioclimatic modeling work. So that's a 30-minute map on the right-hand side there. Now, the 30-minute climatology was quickly followed up with a 10-minute spatial resolution gridded climatology, and we were being dragged to using that for a number of reasons, most of them to do with aesthetics. Because the stakeholders and modelers preferred maps that had this finer spatial grid structure. Now there were several problems with the spline routine in the precipitation layer of the Climate Research Unit data, which meant that there were these patchy, very large errors. The magnitude of the errors was large, but they tended to be sort of very patchy in space. So what was happening was that the way they were minimizing the global fit of the spline came at the expense of some really severe extrapolation errors just in some areas. So one case was Western Tasmania, which is just down here. So that was causing some really frustrating errors with trying to fit the model outliers using this erroneous covariate data. We couldn't sort of struggle to go and say, well, why isn't the model fitting at this point? Try and challenge the model to be able to go and get it to fit, and then finding out frustratingly that the problem was that the covariate data wasn't reliable at that point, wasn't accurate. Okay, now to WorldClim. So there was a group from the CG centres. They were even more ambitious than the CRU, generating a global climatology at not only 10-minute but also 2.5-minute resolution. So it was Robert Hyman's and his team there. So that data set included long-term monthly averages of Tmin, Tmax, and precipitation, but it did not include any measures of atmospheric moisture content, and that's critical for coming up with an estimate of soil moisture. Now, using that data set, you could only produce the first 19 Bioclim variables because they were the variables that relied solely on Tmin, Tmax, and precipitation. Now, the temperature surfaces also included some very large regional scale errors. So, in one example, unfortunately, the emission of a single weather station in central Greenland resulted in a relatively warm subtropical surface over the entire island. So these grids, particularly the 2.5-minute, they were spatially very precise and clearly extremely attractive to modelers, but they were highly inaccurate in places. Now, another issue was that the WorldClim team tried to fill weather station guard adapts as best they could, so I applaud them for this part. Due to the gaps in the station records, they had to relax the time frame limits from which they could include the data. So the standard at that stage was to have a 30-year average spanning the years 1961 to 1990. That was what the Climate Research Unit had done. With the WorldClim team, they had to go and accept some stations between 1950 and 2000 in order to get reasonable length model runs. Oh, sorry, not model runs, observation data runs. Okay, so let's just revisiting our climate data challenge. We needed a data set that was globally conformal, so we couldn't couldn't make use of some of the USA only data sets, for example. We needed a 30 to 50 year monthly average centered on 1975. You know, the inclusion of the 1950 to 1961 data and up to 2000 in the Bioclim didn't bother us because we were happy to go and work with the averages based on 75. They needed to be sufficiently accurate. We didn't want to keep running into the same problems we had with the precipitation layer for the CRU. We needed to be able to go and produce all the variables needed for the Bioclim and CliMax MetMan database. So including atmospheric moisture content to allow the estimation of relative humidity, 9.5, and the soil moisture status. Now that allowed us to then calculate Bioclim variables 20 to 35. So our solution was to create a hybrid data set taking the best quality variables from both Bioclim and the CRU data sets. So from the CRU we took the TMIN, Tmax, radiation and VP, and Bioclim we took their precipitation layers. We had to estimate relative humidity, and we created a soil moisture model in Dymex which took in those variables and combined the results with radiation in order to create the full set of 35 Bioclim variables. Now, undeniably, the availability of data at 10 minutes was an attraction. There's no doubt about that. But we remain a bit skeptical about the value and the meaning of a 30 or 50-year average value for these variables in an ecological sense at a spatial scale finer than 10 minutes. We think we remain skeptical about that. So we developed the CliMond data set at 10 minutes because of the availability of data. There's no doubt about that. And we do acknowledge that finer scale climate sources can pick up this topoclimatic detail in highly dissected terrain. But the results of the study we did here sort of show clearly that it has very little impact on the range of an invasive species, for example. You're picking up details, you're not really affecting the big picture. Now, it seems that routinely producing models using the finer scale data seems to us to be a misrepresentation or misunderstanding of the uncertainty in the climatologies. And that's not even taking into account the compounded errors in the modeling when we apply our modeling tools over the top of those climatologies. Now, this is one of the maps taken from the Hymans et al. Paper. And once again, it reinforces the patchy nature of the station data network. So in some areas, you could sort of mathematically argue that you could drop down to a much finer resolution splined grid. But in others, it seems a bit ludicrous to that in areas such as, say, the Andes or the northwest of Australia, where you've got strong gradients of either rainfall or temperature or both, but very, very little station data. It seems a bit silly to go and have extremely fine scale data. It's sort of misrepresenting what's going on. So just an example of where we used the CliMond data set. We published this paper in 2011 trying to compare correlative models and more mechanistic models, CliMax, in an invasive species context and looking at climate change. Now, in this example, I'm just going to bring to you now, we're only going to look at the invasive species component of it. Now, invasion biology provides us with some opportunities. It's got a challenging modeling problem where you've got to go and deal with novel climates. You've got to deal with invasion lags, where the central assumption of most correlative models that the species is at equilibrium with its environment is clearly not satisfied. And you've got the effects of enemy release. Now, these are topics that that Town and colleagues have explored through their BAM diagram. I won't go into that here at all. Now, invasion biology also gives us some rich data sets to validate models with truly independent data sets. These are data that come from different continents. Now, once again, I think this was a topic that Town wrote about many years ago, suggesting that in order to go and validate data sets, they needed to be spatially biased. You needed independence. Resampling your data or partitioning your data randomly doesn't really test how good your model is when you want to go and generalize it. Okay, so Acacia pigmentha, it's Australia's national floral emblem. It's native in the southeast of Australia, but it's naturalized elsewhere. It's been spread into gardens, and it's spread within Australia. It's become somewhat problematical and invasive even within its native biogeographic region. Okay, it's also been introduced to South Africa, and it's had a fairly long history of spread there. So we were able to go and use the South African data to go and test model projections into South Africa based on the Australian data. What do we want here? So this is just the background on it. It's whilst biocontrol has been moderately successful with a bud galling wasp, seed feeding weevil, it has nonetheless spread as far as we think it's going to, where it's capable of in South Africa. Okay, so here we've got the results of our modelling of the current native distribution with the modelling approaches shown along the top. So we also had boosted regression trees, Maxent, and Climax. I'm only showing the Maxent results here because they were very similar to the boosted regression trees, and just for space and clarity, just showing the Climax. Now the suitability index is from white to purple. The hashed areas represent the novel climate space relative to the background, which is the area in which we need to be cautious with interpreting our projections. Okay, all three approaches captured the native distribution quite well, and a reasonable proportion of the introduced populations in Western Australia and Tasmania. However, it was only the mechanistic model that was able to capture the introductions in New South Wales. So this is New South Wales through here, and actually projected a large area of suitability extending up the east coast up through here. Now this is a high elevation area where you have quite cool, warm temperate climates extending up through here. So they are quite similar to the climate much further south. Okay, so we've got the hashed area here. These are the model results in relation to the distribution points. So we can see here that the correlative models didn't pick this up. Both models picked up the Western Cape and Southern Cape quite well. Sorry, at least the Western Cape. Okay, projecting that globally, now here's where we start seeing a significant difference between the different families of modeling approaches with both correlative models appearing to not get the potential distribution in the cold end well at all, and this has to do with the way they were extrapolating into the novel climate space. Okay, so future climate scenarios. This is one of the elements we've included in the CliMond data set. Okay, we've only included a limited number of emission scenarios and climate models. So we've only got the A1B and A2 scenarios, and CSIRO Mark III and the Miroc High. Now, our rationale for such a limited offering was that the emissions scenarios are just that. They're scenarios. They are insubstantial ideas that were established to be plausible, but they were never intended or capable of being probable. So the set of ESRA's scenarios were never designed to go and be a probability distribution. So considered together, they didn't give you a distribution of likely outcomes. Now, since they were created, several of the conservative emissions scenarios have become implausible. So the B family, yes, we just can't come back to start tracking on that, because our emission scenarios since our emissions tracks since we've created the SRS scenarios have just been far too high. So to include, for example, those B family scenarios and models based on them in in some sort of ensemble is to be disingenuous and misrepresent the likely outcomes. Now, the other thing, coming back to this, what they were designed for, they weren't designed to predict the future. They were designed as a means of stress testing our biophysical models. So it's actually better off going and using more extreme scenarios to go and indicate if there are some sensitivities in our systems that we care about and we need to model. We need to manage. Okay, so some methodological considerations. The baseline climatologies contain these artifacts of topographic and other geographical signals. But the change surfaces are smooth. So when you subtract the long-term average baseline data from long-term global climate model results, the differences they tend to go on very smoothly. So you can spline those down or interpolate those down using very simple interpolation methods. Now, and then you can apply those change surfaces once you've splined them down to the baseline climatologies. Now, this method does, of course, assume stationarity in the nature of the topographic forcing functions. So things like the adiabatic lapse rate, you're assuming that remains, all those functions remain similar. The same with things like rain shadows. Now you can see a good recent paper on many of these issues around applying future climate scenarios by Rebecca Harris et al. Okay, now we didn't just limit ourselves to data. Admittedly, most of the data that was generated by other people, which we're very grateful, and we in our paper and on the website, we urge you to include in the citation the CSIRO and Bioclim citations. Okay, so the Köppen-Geiger climate classes. This harks back to, was it 1936? But hasn't lost its relevance or utility. We've created these, driven mostly by a desire to come up with a rationale to define the background in Maxent and BRT and similar models where similar sort of presence background models. The idea being that you can use the set of Köppen-Geiger classes that your species is found in as the background. Okay, so the classes were derived from consideration of climate patterns and the patterns of vegetation formations. The class rules were derived not from a k-means style mathematical patterning in the climate data, but they would arrive to try to match the observed ecological patterns, so that there's ecological meaning to the classes first and foremost. So we took the class definition rules and applied them to our grid climatologies, and that allowed the topographic signal in the climate to come through into the classification. As we applied the classification, we also discovered that there were some ambiguities in the class definitions, and previous authors had also found some of those. And we had to make some subtle adjustments to remove the ambiguities. Okay, now comparing our results with those of Peel et al., which come out in 2007, and classification differs in that we did include the important covariates in the splining of our climatic data, where Peel et al. Didn't. They just did a simple interpolation. And that's apparent here with, you can see in the CliMond data, we're getting more topographic signals. There's more complexity here compared to the Peel results, which are fairly distinctly smooth. So we're retaining more of the real climate information in the CliMond dataset. Okay, XDET. This is one of our most recent additions, a tool for detecting extrapolation. So it's the bane of it's the bane of correlative modeling. Is when you go into an extrapolation situation that here be dragons, which is the sort of the tagline for the XDET tool. Okay, so in the left-hand side figure here shows the various options for extrapolation in a Maxent model. There we go. We can either retain the last value as we go out to the end, or we can clamp, or we can what's the other one, hinge. Now here, note the ecophysiological response curve, or hypothetical one, we're looking here in black, in the solid black line. The dotted line is the response curve. In Maxent, it tends to be more jagged, more wiggly in BRT models. Now, what else? Okay, so yes, so picking these modes of extrapolation have quite profound differences, or quite profound impacts on how your model behaves when it encounters this novel climate space. But you can see here that these are arbitrarily chosen factors in your Maxent model with extremely different response curves and as we've seen, very, very different impacts. So given the arbitrariness out here, the XDET team were focused on going and saying, well, what we want to know when we get to this spot. We want to know it very, very carefully. Now, the mesh maps in Maxent do this to some extent. The XDET team realized that there were other forms of novelty that weren't being catered to. So there's two types of extrapolation. You've got the covariate range. So this is our covariate along here. There's the first type is when you actually get out of this range. The second type, although we're calling the novelty type 2, is when you've got correlation change, where even if you're within this range, if the relationship between different environmental parameters changes, then you are also encountering a degree of novelty. So in the right-hand side figure, we've got bivariate space here, a series of points within that space, and we can see that one point in the top left here is something of an outlier in that covariate space. Now the mesh analysis, which is what we've done here, works on the range of the covariates min and max, and it would not identify that point as being novel. That is, it's within the mesh space. Okay, now a Euclidean type analysis would also not identify that point as being novel, because it's just on the edge here. However, a Mahalanobis approach does. So this is this oval here clearly defines this point as being an outlier. Okay, so the concept behind the XDET tool, this is the core of it. If this is our training space, so we've got these red dots here, so the red open circles, and we've got this projection space back here in the grey oval. The MES analysis would do something like this. It would put the bounding boxes on. So it would define these the black dots as outliers, and hence be concerned about it. However, the pale blue dots sitting through here represent points where they're within the univariate or the covariate range, but the covariates are differently aligned to each other. So the Mahalanobis distance defines these as outliers and worthy of concern from a modeling perspective. So I'm showing an example here with Acacia cyclops. It's a Southwest Australian native and something of a global invader. Now, I need to point out here that this is our background right here, and this is defined by the Koppen-Geiger zonations. So here the different zones. Now, when we apply the Maxent model, I mean, we get the alien range here is shown as these blue dots, and also here in California. Okay, now we've got unusually high suitability projected for tropical regions, which just seems, from an ecologist perspective, very odd, given that it's a Mediterranean species. So we're figuring that that's a problem with the extrapolation, the extrapolation method that was chosen. Now here with our training domain, remember our training domain is the grey area, and much of the high suitability in the tropics from the previous slide is in that first extrapolation space, type 1 novelty. Okay, it's this red out here. Remember that all of this was showing up as being suitable. Now we're going to say, so we've got ecological relevancy to correlation change is in this blue area here, this bluey purple area. So this is where we're within what you would call the MES map range, but the correlation between the covariates has changed. Okay, here we can see it more clearly. So we need to be skeptical about model results in this area as well. Okay, so XDET has a simple interface and can run on any platform. It exports the results as both an image or a database format, and provides a whole range of data exploration tools, so correlation matrices, box plots, scatter plots, etc. Okay, now one of the outputs is this most influential covariate map. So it's an improvement on Maxent's MOD, or most dissimilar variable map, as it shows both type 1 and type 2 components. Okay, now we also note here that there's considerable niche shift in the model covariates for the alien records. So the green are the alien records, blue are the native. So we're seeing that, that there is substantial niche shift going on. Okay, so with the conclusions around this, this sort of extrapolation of awareness is critical to all modelling, and we see far too many papers these days are being published that ignore or do not quantify model extrapolation for the correlative models. And we hope that this will be something become something of a prerequisite for publication in the future. Now, XTEC can be used to compare a whole range of contrasting raster sets, not just maxent background and projection domains. For example, we can compare climates of the native and non-native records for one species, and so on. The future directions will try to develop some more metrics capturing model performance. There are far more appropriate alternatives to that AUC, and expand the Mahalanobis metrics to model building. Okay, the Bioclim variable registry. So the set of Bioclim variables hasn't always been 35, or some people might have come to an understanding that it was only the 19. Most SDMs that are published these days appear to only consider the first 19 Bioclim variables, and that's probably because they're available in Bioclim at very fine scale. That's because they only rely on temperature and precipitation. By ignoring soil moisture and radiation, I think there's a particularly for plants, and if you consider the plants are primary producers, so even if you're interested in something like an insect, the health of and well-being of the plant host is still going to be a significant, or potentially significant, limit on your potential distribution. Now, considerable recent discussion has been in literature recently about the propensity of modelers to go and use all 19 or 35 Bioclim variables. Just throw them in there, push the button. Now, some people have come up with a pejorative term here, data dredging, where you don't apply any ecological thinking to why you would select certain variables. Just throw the lot in there. Now, it's been demonstrated that this can result in overfitting of your model, and there have been suggestions to ignore correlated covariates as a means of sort of managing this problem, ignoring correlated covariates, and I think more substantially selecting covariates based on ecological relevance. Now, clearly that advice is not being heeded universally, or probably even in the majority of cases. Now, the effects of overfitting are usually not apparent until a model is projected into novel climate space in the new continent or future climate scenarios. All you appear to get is a better model fit when you're just doing a native range species model. So we've just published this recently, where we've decided to go and extend the suite of Bioclim variables using principal components analysis to go and give people a means of dealing with part of that problem. And in doing so, we had to go back and look at what Annuclim was doing with the Bioclim variables, and decided that we could really do with a registry system to allocate numbers to new Bioclim variables as they come out. Now, the first 5 principal components capture more than 90% of the variation in those first suite of 35 Bioclim variables. So it promises a partial solution to the overfitting problem. Now, it might be useful in situations where there's very little known about the ecology of the species and the likely range limiting factors. If you're going to engage in an ensemble modeling approach, throwing lots of species at the model with very little supervision, this may have an application there where you're at least trying to reduce the opportunity for model overfitting. But it should not be seen as a replacement for good, careful consideration of the ecology of your species and trying to go and select variables appropriately to, in terms of what factors are limiting its range. Some limitations that I can think of: it can't be used for climate change studies. as soon as you change your underlying variables, you have to go and redo your principal components. It's purely a descriptive technique. It's likely to be difficult to interpret the inner workings of derived models because the variables themselves are an admixture of the components of the 35 Bioclim variables. Interpreting them isn't necessarily straightforward. Okay, now here's what the first 5 look like when we plotted them out. The first one is clearly a temperature. It's dominated by a temperature signal, and the second by rainfall. The third to fifth start getting a bit more complicated. There's full descriptions of what qualitative descriptions in the paper, in the published paper. Okay, so having come up with some new Bioclim variables, we thought, well, we needed some sort of registry, because we wanted to be able to give them a number so that when people use them, there's a shorthand for going and saying, okay, I use Bioclim, Bio 36, 37, and 38, and that there would be somewhere where there will be a definition of that, so that there wasn't confusion. So the Bioclim variables have traditionally been described in the Bioclim and Euclid manuals, but this wasn't core business for those teams. So some of the definitions were a little imprecise or had changed through time, and the registry wasn't keeping up with them completely. So with their blessing, we've agreed to maintain a registry of the Bioclim variables on the CliMond website. So the way it works, we'll discuss proposals for new variables, and if it's appropriate, if they've got global scope and they're likely to be attractive to a number of modelers, we'll issue new variable numbers and then apply them to the CliMond climatology, if that's appropriate, so that there's an example that's available freely. Now, proponents will also need to go and publish a description and an example of the application of the variable, going in, describing and demonstrating its usefulness. Now, that may be something as simple as just a short note, or something more elaborate. But it's just in terms of keeping in line with our philosophy of documenting material, we thought this was an important component. So the registry is available at that website, and just send us an email if you've got an idea for a new global Bioclim variable. Okay, so the future directions, we want to develop the CMIP 5 future climate scenarios. So they're based on RCPs, or the representative carbon pathways. So the idea there was to move away from the notion of an emission scenario where there was substance. They wanted to go on to make them obtuse, so people didn't treat them with more respect and credibility than they deserved. So, for example, that'd be 2 times CO2 by 2080, and so let's see what that does. We want to develop some more contemporary data sets, so we'll update them. So yeah, things have moved on since 1975. We want to understand better the reliability of the climate data. So we're presently testing the data to go and understand what the uncertainties look like, so that we can pass that knowledge on to the community. We want to collate and share more utilities. So please, if you've got ideas and you want to share them, give us some consideration, and extend the Bioclim variables further. So conclusions, I think modelers, we need to be wary of equating resolution with realism. That getting finer scale data sets doesn't make them any more real, it just makes them prettier. So take care when we're to really consider climate data uncertainty and take it into account when you're presenting models that rely on gridded climate data. The data sets look far prettier than they really are. And yeah, please feel free to contact us if you've got an idea for a project or a new data product. Thank you.
Thanks, Darren. That was great. Believe it or not, the person who requested this, sorry, there's no applause, but I'm not good at doing that because I'm alone in my office here. The person who requested this lecture, Clement Mwea in Tanzania, was up watching you. So that would be, let's see, 9 hours, something like 2 in the morning or 3 in the morning. So,
oh.
so the opposite problem that you have. And so what he's written as a question is, I'd like to thank you so much for this important lecture. I'm following it very closely. I will be really happy to try running these variables and compare predicted results for my previous outputs for Rift Valley fever and dengue vectors in Tanzania. I feel like these added variables can bring a significant difference in distribution of disease vectors as they are highly influenced by atmospheric moisture. Can Professor Kriticos share these new variables that I can test using MaxN? I'm having difficulties in getting them from their website, and the format is different from what I am used to as .asc files. That's the first question. You want to maybe take that on, Darren?
Sure. Okay. Sorry, I need a point of clarification because we provide the ASC files. They may have a .txt extension, but you can find those files, the raw data files, in, I think it's called raw climate. If anyone's got problems accessing data like that, if they send us an email contact on the website, one of the team will get back to them within about 24 hours, and we'll make sure you get the data.
Wonderful. So those .asc or .txt files, those are classic ASCII raster grids?
That's correct.
Okay. So also, Clement, if you have trouble with that, let me know and I can walk you through that. It should be pretty straightforward. Second question from Clement is, this will be an interesting one. How are these future climate scenarios different from what you, which is to say I, provided to them in the course that we did in Nairobi in 2013? Were they from the A1B or A2 series? From this lecture, do we need to avoid using these scenarios because they were not developed for the purpose of predicting future distributions? So,
okay, so do you want to handle the first part of that, and I'll handle the second?
Exactly. So the climate data that we gave you all in Nairobi in 2013 were data that were based on the WorldClim data set. So essentially the current climate was estimated using the techniques that Dr. Kriticos just went over, but as part of the WorldClim effort. The future scenarios were keyed to those present scenarios. So essentially there's a method of downscaling called a delta or change method, where you find out by subtraction within the model results the change between current and future. You smooth that, you add it to the current to get a smoothed estimate of the future. And what the smoothing does, in effect, is downscales the future, which initially is at very coarse resolution, to the resolution of the present data, which are quite a bit finer. So we gave you future climate data that were based on the WorldClim data set, and what you're seeing on CliMond is based on the CliMond data set. That an adequate response, Darren?
Yes, that takes care of the the methodological components. Now the epistemological part of it, in none of those cases, Clement, do we have predictions. The data that town supplied you and the data that we're supplying you, they are not predictions of what will happen in the future. Okay, so if you build a model based on them, you are not building a model that predicts the distribution or the potential distribution of your organism in the future. Okay, the atmospheric scientists don't have a crystal ball that looks into the future to go and say what was the climate like in 2070. Okay, what they've done is taken a series of storylines. These are fictional storylines intended to be to sound as though they could happen. Now, their purpose wasn't to go and predict the future. Their purpose was to go and say, well, if these different scenarios did play out, what would the future look like? So the term we try to use there to go and emphasize this is that we call them projections. That if a certain set of hypothetical circumstances were to happen, our model would project that this is the consequence. Now, the way you should be using them in a species distribution modeling context or a niche modeling context is to go and then say, okay, if that future climate happened, if our organism experienced that future climate, where do we think it might, its distribution might fall? Now, the purpose isn't of that exercise, or a legitimate purpose for that exercise, isn't to go and say our species will get to this point at that nominated point in time. What we want to do is go and use it as a means of stress testing the system that we're managing. So if it's an invasive species, okay, could that invasive species reach this area that I have to manage in that future climate scenario? So should I be concerned about it in the future? On the flip side, if it's a species of conservation concern, if I was to apply, we look at this future climate scenario and we go and see that the range of that species, or the potential range of that species, appears to have shifted outside of all of our reserves, then we might think, okay, we're going to have to monitor that. Now, the appropriate management response isn't to then go and start getting reserves organised in those areas that that start lighting up as suitable. What we do then is put in an adaptive management framework to go and see whether that future, which of those future climate scenarios is transpiring, and start thinking about measuring variables that tell us that we might need to go and start organising reserve systems that are aligned along that gradient of potential change. It's a challenge, and so many people and so many papers have fallen into the trap of looking at that future data as though it is a prediction. So that's a long way of coming to the point to go and say, well, it actually doesn't matter terribly much whether you use those WorldClim scenarios or the scenarios we provide. It doesn't really matter, but make sure that you're using some of the extreme scenarios, because otherwise, if you were to use a B series, you're not really stress testing the system, and you're not stress testing it in a manner that's plausible anymore. Sorry for that long-winded.
No, that's perfect. Thank you. Here's a second question. This is from Monica Papic, who is a doctoral student here 5, 6 years ago and is now an assistant professor at Oklahoma State University. And she asks, will CliMond future climate data sets be updated to include the IPCC 5th assessment of emissions in the RCP scenarios?
Okay. That one's got a short answer is yes. So that's the CMIP 5 scenarios, and I'm working on that sort of with Rebecca Harris from the Cooperative Research Center, the ACE CRC. Oops, I'm sorry, Rebecca. She may be watching this, but I can't remember what the ACE is. It's Antarctic climate change or something like that.
Okay. And then we have a third, this is just a comment more, but it's funny enough that I have to pass it on to you. Let's see, hold on 1 second so that I can read it to you. Okay, great talk by Darren. Kriticos, new bioclim layers and some important cautions on predicting species distributions to novel climates. Last sentence, time to step across the road for a chat. Don Rosauer. So this is a very close neighbor of Dr. Kriticos, into a global seminar and now interested in this body of people. So that's probably an opportune place to stop. Thanks a lot, Darren, for an hour of your very early morning. And everybody else, tune in next March 26th for the next seminar. Darren, many, many thanks for your time.
And thank you, Town, and thank you, everyone else. Keep well.
Have a good day.
Ciao.